Hey guys, welcome to AP Biology, and we're going to do a quick lecture on nucleic acids, which deal with information transfer and storage. So please pause and stop this video whenever you need to look at your notes and write something down if you need to. If you have a question, please do that. That is extremely important. All right, so let's go ahead and see what we got to do. So we've already talked about three of the categories of biological macromolecules. We've talked about carbohydrates, lipids, proteins, and today we're going to focus on nucleic acids. Again, they are the molecule of information, the molecule of heredity. They include both DNA and RNA, and the interesting thing about DNA is it can copy itself. And the main function of DNA is to transfer information from one generation to the next. And do remember that information transfer is one of the big ideas that we see in biology all throughout whatever we study. And this is probably the biggest. Okay, and I have a little DNA from the beginning diagram on the picture on the bottom. GIF, we see a sperm cell. And again, example of that DNA being transferred from one generation to the next with sexual reproduction, but also through asexual reproduction. The first thing that has to happen anytime a cell divides is that the DNA must be copied first. DNA also plays a role in directing RNA synthesis, and again, through that process, controls protein synthesis. So this is a very important molecule. So let's look at what that flow of information is. You're going to see this multiple times, but I'm introducing it early to see the role of DNA and the role of RNA. And I should say one of the roles of RNA because there are many, many different types of RNA. And we're going to focus on basically about 5 of them in this particular class, but just be aware there's a lot more. But anyways, the flow of information is from DNA stored in the nucleus in eukaryotic cells to RNA in the form of messenger RNA, which basically carries the message, which is the instructions for building a gene, out to the site where the genes will be turned into a protein, and that will be a ribosome, which would be in the cytoplasm of a eukaryotic cell. Okay, so the messenger RNA, the mRNA, does play an intermediate role in shuttling that information for a single gene, which remember in this case would be the instructions for assembling a sequence of amino acids, which is again which level of protein structure. Okay, I'll let you answer that particular question. All right. So nucleic acids, just like other polymers, are polymers. Okay, they are polymers. They are immense polymers. And the monomer that makes up the nucleic acids are nucleotides. Those are the building blocks of both DNA and RNA. And each nucleotide is also somewhat of a little tiny polymer because it is made up of 3 basic parts. We have a phosphate group, we have a 5-carbon sugar, and then we have the nitrogenous base. Okay, and there are 4 different DNA nitrogenous bases and 4 different RNA nitrogenous bases. One base, thiamine, is unique to DNA, and another base, uracil, is unique to RNA. Do notice that the sugars are slightly different in DNA and RNA. In DNA, it's deoxyribose, and in RNA, it is ribose. And so again, the name for DNA, that's an abbreviation, stands for deoxyribonucleic acid, and for RNA, it is ribonucleic acid. And D means to take away. And if you notice, there are 2 hydroxyl groups on ribose, and there's only 1 hydroxyl group on deoxyribose, okay, because 1 was taken away. That's what that word deoxyribose means, take away one of those sugars. Or hydroxyls, excuse me. DNA is a double-stranded molecule. It consists of two strands of nucleotides that are running in opposite directions. What does that mean? Well, look at this picture closely, and you can see that this strand right here on the left-hand side, we have the phosphate on top, and we have this OH group on the bottom, where the opposite strand on the right-hand side is we have the phosphate on one end and we have the OH on the other. We say that these strands are anti-parallel and complementary to each other. What does that word complementary mean? Complementary means that one base will always pair with another base. Thymine always pairs with adenine in DNA, and guanine always pairs with cytosine in DNA. Also notice some of the other things between the thymine and adenine bonds. There are nucleotides. There are 2 hydrogen bonds, and between the guanine and cytosine, there are 3 hydrogen bonds. So there's some differences there. So we'll go through and look at some of these in a little bit more detail. So anti-parallel means they are running in opposite directions. Complementary means that if you have one strand, because of these base pairing rules, T to A and G to C, then we can always build a complementary strand that is exactly that will lead to 2 new molecules that are exactly the same. So each strand can act as a template, if you will. Okay, so let's dive a little deeper into the structure. So when we look at a nucleotide, once again, we can see that there's adenine and guanine. Those are what we call purines. They have this double ring structure. There's one ring, there's the other ring. There's one ring, there's the other ring. Adenine and guanine are found in both DNA and in RNA. And then we have another structure, pyrimidines. Pyrimidines are single ring structure. Cytosine, one ring. Thymine, one ring. Uracil, one ring. And remember, uracil only found in RNA. Thymine only found in DNA. And again, there's our deoxyribose sugar on the bottom and are compared to our ribose sugar. And so we have to have one phosphate, one sugar, and one nitrogenous base to make up a nucleotide. And this is a picture of a polynucleotide over on this side here. Okay, so the structure of DNA, the discovery, we'll dive into that in Unit 6, was a long-running story. It took a long time to figure out the structure of DNA. DNA was first discovered back in the late 1800s. All right, so it took almost 75 years or so before the structure, how DNA is actually built, was put together, what it was composed of. We knew that in the late 1800s that there were these nucleotides, but we didn't really know how they were put together, or we didn't really know how the 2 strands, or even if there were 2 strands. It wasn't until, once again, Watson and Crick in 1953 published a paper that basically wowed the world and began the DNA revolution, which we are well into at this point. Even though we're well into it at this point, talking about 75 or 78 years later, we still have a lot to learn. So if you're interested in this kind of stuff, there is room for you to discover a lot more. Okay, so we already mentioned the hydrogen bonds. Okay, between, and also the other thing I want you to know, A always pairs with T and G always pairs with C. What else do you know? A purine, adenine or guanine, always pairs with a pyrimidine, which is cytosine and thymine in DNA, or cytosine and uracil in RNA. Okay, and what does that do? That keeps the width of the 2 strands constant, because think about it, if we had 2 purines, 2 double ring structures paired with each other, the strands would be far apart, and then if we had 2 pyrimidine single ring structures paired with each other, the strands would be really close together. So by having an adenine and thymine always pairing, then that keeps those 2 strands at a constant distance apart. And you need to know the differences between DNA versus RNA. Pause the video here, look at those, make a note of it, and make sure you know what those differences are. Okay, once again, this is a polymer that is built using dehydration synthesis and broken apart by hydrolysis, just like every other polymer. Now, one thing, DNA does have direction, okay? And what we're looking at right here, here's my phosphate. This is a nucleotide, but we're not seeing the nitrogenous base, which would be off on the right-hand side here. What we see is the phosphate, and we see the sugar molecule. And the 5-carbon sugar, each one of these vertices of this molecule is a carbon molecule. You don't always see that, but there's a carbon there. And we number the carbons clockwise, starting with the carbon that is attached to the nucleotide, to the nitrogenous base. I should say attached to the nitrogenous base. This is carbon number 1. This is carbon number 2, carbon number 3, and carbon number 4. And carbon number 5 is up here and attached to a phosphate. So we can only add new nucleotides to what we call the 3' end. That's what that little thing there means, prime. So this is the 3' end where the hydroxyl group is, and this is the 5' end where the phosphate molecule is. And this causes some problems during DNA replication. And again, that nucleotide sequence is extremely important. What do I mean? That's the sequence of A, T, C's, and G's. The only difference between you and me is that sequence of nucleotides. The only difference between me and a carrot is the sequence of nucleotides in our DNA. Everything else is basically the same in terms of the DNA and how we go from gene to protein. It's the same in a carrot, it's the same in a bacteria, it's the same as you and me. Okay, again, another piece of evidence for that common origin of all life forms on the planet. But again, that nucleotide sequence, the sequence of A, T, C's, and G's is extremely important. And if that sequence is off, as we've already discussed about sickle cell anemia, that can lead to a different amino acid, which can change the shape of a protein, which can be a life-threatening disorder in many cases. Okay? Once again, inheritance is based on the replication of DNA double helix, and it's based on the faithful replication of that DNA double helix. Okay, we have the base pairing rules: A pairs with T, G pairs with C. And if these strands, when these strands are split apart prior to DNA replication, each strand now acts as a template for a new strand. So because of the base pairing rules, when you separate those two strands and allow them to be paired with new nucleotides, you will end up with two copies of DNA that are identical to each other. Each DNA molecule will have one old strand and one newly built strand. And we'll get into that again in Unit 6. Okay, we can use DNA, changes in DNA and proteins, as a measure of evolution. Again, the molecules are passed from parents to offspring. Therefore, your siblings have a greater similarity than do unrelated individuals of the same species. So these are my 3 kids quite a while back, and that's the gravesite of both my parents, who died relatively young. And this is in my right actually across the street from where I grew up. And there's a graveyard, and it's a real pretty little graveyard surrounded by cow pastures and whatnot. But my genes were passed from both my mom and dad. We were passed from their mother and father, my grandparents, and all the way, just keeps on going back and back and back further. And as you guys probably know, that you may resemble one of your grandparents or so, or maybe even a great-grandparent. And again, so the more closely related you are, the more similar your DNA is. Therefore, we can even use this as a yardstick to compare us to other species. Once again, our closest living relative is a chimpanzee. Did we evolve from chimpanzees? No, we didn't. We evolved from a common ancestor that was neither chimp nor human. And over the 7.5 million years since that split has occurred, chimpanzees have evolved into many different species, and finally to what we have are, the common. Basically, I think we have about 3 species of chimps right now. Humans evolved into a multitude of different Homo species, but those species have all gone extinct except for us. We're the sole remaining Homo species out there. Okay, so the more similar the DNA is between two species, the more recent their common ancestor was. We share 60% of our DNA with carrots. When was our last common ancestor? Well, that's probably closer to 600 million years ago. We had a common ancestor a very long time ago. All right, so we can use this, and we'll get into this a lot more in Unit 7. Okay. And there's my kids now. This was actually a few years back when my youngest son in the middle there had his white coat ceremony. Oh, no, that was when he got matched with his residency program. And my oldest son on the left, that's Ryan, and my daughter, Ilya, who's at vet school at Auburn. Well, thanks for watching. I hope this helped.